Hi, my name is Marie Attard and I'm a postdoctoral research assistant in Steve Portugal's lab at Royal Holloway, University of London. Today I'm going to be talking to you about how climate change and parent nesting strategies influence gas exchange across avian eggshells. Birds have evolved multiple functional traits to improve offspring survival in the nest. Arguably one of the most important is the eggshell. Eggshells are covered with millions of microscopic pores, allowing the embryo to breathe through the diffusion of gases such as oxygen, carbon dioxide, and water vapor. Eggshell conductance is defined as the rate of diffusion of gases through the shell and can be easily quantified by assessing mass loss of the egg over time under controlled temperature and humidity conditions. Most bird species lose between 10 to 20% of their fresh egg mass over the incubation period for healthy embryo development and hatching. Eggs that lose too much water during incubation frequently do not hatch due to desiccation, while embryos that do not lose enough water from the egg experience respiratory problems or drown. Maintaining controlled loss of water from inside the egg to the external environment is therefore essential for embryo survival. Birds are highly diversified and widely distributed, occupying every continent on Earth and every terrestrial habitat within it. The optimal degree of water vapor conductance of an egg varies markedly across species, which can be partially attributed to their nesting environment. Predicting eggshell conductance of a species is not always straightforward, as multiple ecological factors must be taken into account. Brood chambers of burrow nesting species, for example, are permanently saturated with water, which promotes the evolution of high eggshell conductance. Burrow nesting bird species, however, tend to have a long incubation period, which would promote the evolution of low eggshell conductance. Consequently, knowing the differences in eggshell conductance of a species can only be determined by considering the contribution of multiple life history traits and the phylogenetic history of the lineage. The aim of our study was to determine to what extent does phylogeny and life history influence eggshell conductance across modern birds. Previous studies, mainly from the 1970s and 80s, typically focused on either one group of birds, such as gulls, with the goal to look for micro-adaptations between closely related species, or to look at eggs of extreme nesters, such as desert nesters, water nesters, and marsh nesters. In 2014, Portugal and colleagues attempted to tackle the challenge of understanding what factors drive the evolution of eggshell conductance using a large-scale comparative analysis across non-passerines. They found that species where parents returned to the nest with wet plumage had a significantly higher eggshell conductance value compared to species that returned to the nest with dry plumage. They also found that birds that typically breed on cliffs, in ground burrows, or in tree cups had a significantly higher eggshell conductance value compared to species that breed in tree cavities, on the ground, or in tree shallows. Overall, this suggests that the humidity of the nest environment itself is particularly important in predicting eggshell conductance in non-passerines. But what about the passerines? Passerines are songbirds or perching birds that account for 60% of the 10,000 living bird species we see today. All passerines are land birds that have altricial young, whereas non-passerines include both land and water birds that can have either altricial or precocial young, depending on the species. There is also a third group called the near passerines that include sandgrouse, cuckoos, pigeons, hummingbirds, swifts, and nightjars. Originally, near passerines were thought to be more closely related to the passerines due to ecological similarities, with all the near passerines except the sandgrouse being land birds and having altricial young. However, today we actually know that the near passerines are part of the non-passerine group due to genetic similarities. In this study, we decided to to use these free groups to assess whether neopasserines have more similar conductance values to the passerines versus the non-passerines. In our study, we used eggshell conductance values measured from whole blown eggs from 365 species spanning 28 of the 49 avian orders. We directly measured eggshell conductance from over 2,500 empty eggs kindly provided by the Natural History Museum in Tring. We also incorporated mean species eggshell conductance values reported in the literature. To measure eggshell conductance, we filled these empty eggs with distilled water and then sealed the blowhole with some glue and a bit of plastic. The eggs were then placed in a desiccator at 0% humidity and 30 degrees Celsius. The eggs were then weighed every 24 hours over consecutive days to ensure the amount of water that's being lost remained consistent over time within an egg. The reason why we used distilled water was because it actually has no difference in mass loss of an egg compared to if you filled up the egg with albumin and yolk. Phylogenetic signal in eggshell conductance was measured using Pagel's lambda to determine to what extent related species were more likely to share similar conductance values than species drawn randomly from a tree. Pagel's lambda was found to be very high for eggshell conductance before accounting for differences in body mass, but was intermediate after accounting for differences in body mass of the species. This showed that eggshell conductance across birds is influenced by phylogeny as well as other evolutionary processes such as life history, particularly after accounting for differences in body mass of the species. We use phylogenetic generalized least squares models to test the evolutionary association between eggshell conductance and life history traits within a phylogenetic context. We incorporated 14 key life history traits as predictors in PGLS models that had previously been hypothesized to play a role in the evolution of eggshell conductance. So how this basically works is that we ran a series of PGLS models with up to 5 predictors per model. We then applied conditional averaging to predictors that were present in our top-ranked models, which was defined as any model that had an AIC value of less than 2 compared to the model with the lowest AIC value. Using conditional average modeling, we then were able to determine which of these predictors were most important in assessing eggshell conductance across all modern birds. The phylogenetic tree shows the relationship between eggshell conductance of whole eggs across the 364 bird species included in our study. Branch colors show species with high eggshell conductance in blue and low eggshell conductance in orange and red. The body mass of the species is shown in the green bar plots, and in the outer ring we have the non-passerines in dark blue, neopasserines in light blue, and the passerines shown at the bottom in pink. We found that non-passerines had a significantly higher conductance value than that of the neopasserines and passerines, both before and after accounting for differences in adult body mass. This is further emphasized in the bottom graph, which shows eggshell conductance on the y-axis and adult body mass on the x-axis. This demonstrates that neopasserines have more similar eggshell conductance values to passerines, which they are more ecologically similar to, compared to that of the non-passerines. Similar to the previous graph, this figure plots eggshell conductance as a function of adult body mass. Each dot represents a separate species, and the minimum convex hull is plotted for all species within a subset of orders, with each color representing a specific order. As expected, eggshell conductance was significantly higher among heavy species, since initial egg mass increases with body mass. To account for differences in adult body mass, we used the residuals from a PGLS regression of log conductance and log body mass as our response variable. Using this second series of PGLS models, we can ask how well does one or more life history traits result in higher or lower eggshell conductance than is expected for a given body mass of the adult bird. This study is the first to identify a broad-scale reduction in eggshell conductance across modern birds where temperature seasonality increases. And this was found to be true both before and after accounting for differences in body mass. Regions of greater temperature seasonality experience a greater range in temperatures over the course of a year and correlates with an organism's temperature tolerance breadth. Avian embryos can adjust their metabolic activity in response to varying environmental conditions, and by doing so, alter their period of development. The low metabolic rate expected for embryos incubated in highly seasonal climates would favor reduction in eggshell conductance in order to prolong their incubation period, as suggested by our findings. We also found that eggshell conductance was significantly higher in species where both parents incubate the clutch, after accounting for differences in adult body mass. Shared incubation between two parents allows one of them to be relieved from incubation to feed, while the other one incubates the egg. This allows the egg to be covered at all times. Clutches that are incubated by both parents therefore encounter less variation in eggshell temperature than clutches that are incubated by a single parent, and therefore are expected to have higher eggshell conductance values for their body mass. Overall, eggshell conductance across birds is primarily influenced by phylogeny, body mass, temperature seasonality, and parent incubation strategies. This study highlights the remarkable functional qualities of eggshells and their importance for embryo survival in extreme climates. I would like to thank Douglas Russell from the Natural History Museum for providing eggs for our study. I would also like to thank Scott Partridge, the Western Foundation of Vertebrate Zoology and Philopic, for providing images for our presentation and for our paper that was published this year in Proc B. And lastly, to the Leverhulme Trust for funding our research. Thank you for your attention.